All right, so our next topic are going to be the basic components that are associated with viruses. Now, in terms of viruses, there are going to be two components that are going to be found in all viruses, okay? One of those components is going to be the genetic material, all right? Now, what's unique about viruses? The genetic material for viruses is either DNA or RNA. All right? This is something else that's unique with viruses. Cells are going to have both DNA and RNA associated with them. Viruses have one or the other. There are some RNA viruses, like the HIV virus. There are some DNA viruses, and in terms of that, the genetic material has to be one of those two. There aren't any viruses, at least that have been identified so far, that have both RNA and DNA in them, okay? The second component, oh, this is another squeaky marker, is going to be the capsid. And this is going to be the protein component of our viruses, all right? And in terms of those capsids, they're made up of capsomers. I think I misspelled that. I don't think there's another E there. Capsomers. All right? And in terms of that, the capsomers are the individual units of protein. The capsid actually makes up the entire coat around the virus. So if I draw a virus here, I'm going to make my squiggle here. We'll say this is a DNA virus, just for fun, okay? I'll draw line around that. This is our capsid, okay? The capsid is what surrounds the DNA in the viral particle, all right? The capsid is going to be made up, I put some little dots here, of those capsomers, all right? And those capsomers are going to be the individual protein, all right? So at minimum, this is what you need for a virus. You need the genetic material and you need a capsid, okay? Now in terms of that, there's going to be components that will be present in some viruses, but not all, all right? Again, viruses are relatively simple, so we don't have a whole lot of components here for you to memorize, all right? Some viruses are going to be what's known as enveloped viruses, okay? Which means there's a cell membrane surrounding that virus, all right? So if I draw blue circle here, this is our envelope. And that envelope is essentially a membrane, all right? Provides another level of protection for the particular bacteria. Second part that will be present in some bacteria but not all are going to be the presence of spikes, okay? And these typically are glycoproteins. I'll just kind of draw them randomly here off our virus. The spikes typically are going to be involved in recognition for the virus. So the spikes a lot of times are going to be that component of the virus that's going to attach to or recognize the host cell so it can kind of latch on. All right? So the spikes are found in some but not all. Envelope is found in some but not all. And in terms of our viruses, all right, really they fall into two categories. We've got naked viruses, and we've got enveloped viruses. All right? Naked viruses just have the capsid. Okay? So in terms of that, if I draw one here, this is with our DNA. And our capsid, this would be a naked virus. All right? The virus I drew up here, this, because it has that envelope, because it has that cell membrane, this would be an example of an enveloped virus. Okay? Now in terms of our naked viruses, all right, our naked viruses can also have spikes associated with them. The spikes don't necessarily have to go with the envelope, all right? But not all viruses will have spikes. Not all viruses will have envelopes. All viruses will have capsids and will have genetic material, okay? In terms of those capsids, just to go a little bit further on those, you've got essentially, I'll do this as an A and a B, okay? You've got two types of capsids that are present in viruses. You've got a helical capsid, which I'll kind of draw as dots here. You know, will kind of form essentially like a tube. All right? So that helical capsid is going to be kind of rod-shaped in structure. And that rod-shaped structure is usually hollow. Inside that hollow tube, that's where your genetic material is going to be. Okay? The Ebola virus is an example of a virus that has that helical conformation. All right? You can also have capsids that are known as icosahedral. And essentially, those icosahedral capsids are kind of more triangular in shape. And what you've got typically with the icosahedral capsids is you've got 20 or so of those triangles that come together to form the capsimer there. All right? And in terms of those icosahedral type viruses, adenoviruses are an example of viruses that have that icosahedral. Some of your common cold viruses are types of adenoviruses. All right? Helical, the example I gave you there, the Ebola virus is an example of a helical. All right? So in terms of that, those are the two more common types of capsimers that are associated with viruses that will infect humans. All right? There is a third classification of capsimers that we'll talk about, or arrangements of the viruses that we're going to talk about. But those are going to be what's known as bacteriophage. And bacteriophage are viruses that will specifically infect bacteria. So they're a little bit different. And then my next video is going to go over bacteriophages and how bacteriophages infect bacterial cells. Okay? So viruses really don't discriminate. Eukaryotic and prokaryotic cells can be infected with viruses. You've got different types of viruses that infect a eukaryotic cell. Typically, those eukaryotic viruses are either going to have a helical or icosahedral type capsimer. Bacteriophage are a little bit different, and I'll talk about those in the next video. If you have any questions, please let me know.